Now we will begin our review of chapters 4, 5, and 6 in preparation for our second exam. In chapter 4, we talked about cells. As you recall, smaller cells have a greater surface area. As cell size increases, as cells get bigger, the volume inside the cell is going to grow more rapidly than the surface area of the cell. When we talked about the theory of endosymbiosis, that's when we talked about how two prokaryotic cells ended up engulfing each other and one lived inside another prokaryotic cell as part of evolution. This is believed to be a precursor to the development of more complex eukaryotic cells. Organelles that are believed to have originated from the combination of two different prokaryotic cells are mitochondria and chloroplasts. Okay, when we're talking about the difference between prokaryotic and eukaryotic cells, the biggest thing is going to be that eukaryotic cells have a nucleus. It's where they store their genetic information, right here. They also have internal membrane-bound compartments, so you can see all of these organized compartments that are within a eukaryotic cell. They are membrane-bound. It's very complex, and so we have to have more structure for our function. In prokaryotic cells, you can see we do still have genetic information. We have DNA, usually circular DNA, and it's not bound within the nucleus. It's not contained in any specific way. It's just present inside the prokaryotic cell. So prokaryotic cells have no nucleus. Eukaryotic cells do have a nucleus and multiple internal membrane-bound compartments. All bacteria and archaea are prokaryotic cells. That makes it easy to remember which of these two cells are the simpler cells. Prokaryotic cells are simpler. All bacteria and archaea are this type of cell. Also, prokaryotic cells do still have ribosomes for protein synthesis. They have a flagella that can help them move, and they have pili, which are able to increase their surface area and enhance genetic information exchange. All organisms other than bacteria or archaea are eukaryotic cells. So most of the diversity in our world is going to be made up of eukaryotic cells, the more complex cells that have a nucleus and have membrane-bound compartments inside the cell. You can see that prokaryotic cells don't have a membrane-bound nucleus, but they do have DNA, and that DNA usually bundles together. It's circular, and that area that it usually bundles together in is referred to as the nucleoid region. The ribosomes within the prokaryotic cells make proteins. The pili help attach to substrates and exchange genetic information, and the flagella allows cells to move around. Now here are more complex eukaryotic cells. We have our nucleus, where we're storing genetic information. The nucleolus makes the ribosomes. Here are our ribosomes that have been made inside the nucleolus. The nuclear envelope is the membrane surrounding the nucleus. The endoplasmic reticulum is responsible for making proteins in the rough ER. You know that it's the rough ER because you can see the presence of ribosomes on this part of the ER. So this is the rough endoplasmic reticulum where proteins are made. The smooth endoplasmic reticulum is also making things, but not proteins. The smooth endoplasmic reticulum is making carbohydrates and lipids. As we discussed, our ribosomes are making proteins. Mitochondria break down glucose and make ATP. That's why they're called the powerhouse of the cell, because they're making the ATP that powers cellular activity. And the Golgi apparatus is what we talked about being like UPS. It's basically there to help package and distribute proteins and lipids from the ER. So we've got great proteins coming from the rough ER and carbohydrates and lipids coming from the smoothie are, but those need to be packaged in a way that is receivable wherever they need to go. And so that's why they go from the ER to the Golgi apparatus. That way they can be rearranged, packaged, and then distributed where they need to go. The cytoplasm isn't really shown here, but it's just basically the semi-fluid matrix that everything within a eukaryotic cell is floating in. So that is filling the entirety of the cell. Okay, we talked about resolution and that most cells are too small to be viewed by the naked eye, which has limited resolution. When we need to increase our resolution, we use a microscope. We increase our magnification. And resolution is just referring to the minimum distance that 2 points can be apart and still be distinguished as 2 separate points. So basically, the resolution, if you're looking and you think you're only looking at one person in a picture, and then you increase the resolution and you see, oh, that's actually 2 people at a distance, but they're standing about 5 feet apart. That is just an example of resolution. And that's what we're increasing when we're using magnification through a microscope. Okay, we have bulky substances that are contained within vesicles as they are moved into and out of a cell. Whenever we're doing this and we're bringing substances into the cell, that is endocytosis. It is the engulfing of substances outside of the cell in order to form a vesicle that is brought inside the cell. So you could see here, we've got something that wants to come inside the cell. It's snuggling up to the membrane, it is being engulfed and then encapsulated, and it has been brought into the cell. Endocytosis. In exocytosis, it's the opposite. That's when we have something inside the cell that we want to get out. And those molecules, those secretory vesicles, will again approach the lipid bilayer so that a connection can be made and an opening can be formed and the vesicles can be expelled from the cell. Exocytosis. Endocytosis, you're bringing something into the cell. Exocytosis, you're taking something out of the cell. In eukaryotic cells, DNA is divided into long segments called chromosomes. So here you can see a somatic cell with a nucleus containing genetic information. That genetic information, DNA, is bound up and packaged into chromosomes. So when we're talking about chromosomes, we're just talking about the nicely packaged units of DNA. Okay, plant cells and animal cells. Anytime you're trying to look at 2 cells and differentiate if you're looking at a plant cell or an animal cell, you could look for the 3 things that are not in animal cells. Plant cells have this central vacuole, which we discussed is useful for storing water and starches, which plants do a lot of. They're constantly making starches to store, and if you overwater them, they have to have some capacity to hold that water. So that's the central vacuole. You can remember it usually looks like a swimming pool. Also, plant cells have chloroplasts. Animal cells do not. Chloroplasts are useful for photosynthesis. So all of our photosynthetic processes are happening inside the chloroplasts. It's worth noting that some bacteria and algae that are photosynthetic also have chloroplasts, but not animal cells. Also, plant cells have a cell wall. This is what keeps them from bursting as easily as animal cells do whenever they are filled with too much volume. An animal cell would be more likely to lyse, where a plant cell has this structure from the cell wall that really strengthens it and keeps it from bursting. In summary, plant cells have central vacuoles, chloroplasts, and cell walls. Animal cells have none of those things. When we're talking about our vacuoles, we have the central vacuole that we just discussed, useful for storing water and starches in plants. And then there are also contractile vacuoles, and these are useful in multiple scenarios, but the basic point is when you have too much of something and you need to expel it, these contractile vacuoles are able to pump excess water from inside the cell and pump it out. So this is another mechanism to prevent a cell being too full of liquid, a contractile vacuole. Okay, we discussed molecular motors and how to get a vesicle a long distance across a cell. There are useful components to this that all work together, and the best metaphor I could think of for this is just a train. So if we've got our cargo that needs to be carried, that's our vesicle. Wherever this needs to go, it could be hormones to trigger ovulation, it could be something for your immune system to fight off a cold. Whatever needs to be carried is the vesicle. Then we have our connector proteins. These are going to act like the cargo cars of a train. You can see here, they attach to what needs to be carried. So they are holding whatever needs to be carried, just like the car of a train. The car of the train has to have wheels that connect to the track, and that's what we could think of as our motor molecules, like the wheels of a train. So they are attaching to the cargo cars and the train tracks that are leading to the destination. These are the microtubules. So you remember seeing that super complex web of microtubules going throughout the cell? That's what's helping a very specific molecule get to its very specific destination without getting lost in all the mumbo jumbo that's happening at all times during cells, during all of the cell metabolism and everything that's going on. We need a direct route, and that's what the microtubule provides. Okay, so how do we get rid of worn out parts of cells? Cells are constantly being turned over and digested and broken down into parts that can be reused. Lysosomes are the organelles that contain enzymes to help digest worn out parts of cells. Very similarly, we have peroxisomes, and it can be confusing to differentiate because they sound so similar and their functions are very similar. They're both breaking down things that we don't need. But lysosomes are targeting digestion of worn out parts of our own cells. Peroxisomes are trying to break down oxidants and potentially harmful molecules. So my hint for remembering this is just peroxide is often used as a cleaner around the house, and peroxisomes are for cleaning out harmful molecules in the cells. So lysosomes, you're breaking down worn out pieces of your cells, and peroxisomes are breaking down oxidants and potentially harmful molecules. Whenever we're discussing the fluid mosaic membrane, remember that's just this image that is showing you a representation of the phospholipid bilayer and all of the different parts that are present within that membrane. So you can see we have our integral proteins, our glycoproteins, a peripheral protein, and the fluid part of this is just meaning that none of these things are locked into their place just because you see them this way in our 2D image. The fluid part is to remind you that all of these pieces are moving. They are not locked into one specific location within the membrane. This protein could move and suddenly be over here at some point. I included these pictures of the mosaic just so you could remember that when we're talking about the mosaic, the fluid mosaic membrane, or the fluid mosaic model, that's the representation of the membrane. And we call it mosaic because you're able to see all the individual pieces just like you are with mosaic art. We have our overarching picture, but you're able to see all the individual components that make it up, and that's why it's useful. So when we're talking about our membrane of our cell, that's made up of the phospholipid bilayer. When we zoom in and pick at one piece of that phospholipid bilayer, one component that's making up the whole membrane, it is this structure, a phospholipid structure with a polar head and two nonpolar tails, as seen here. Okay, so movement of water and nutrients into a cell or elimination of waste out of a cell is essential for survival. We constantly need to get water in, we need to get nutrients in, we need to get wastes out. Those things happen in three different ways. There are three ways to cross the biological membrane. It's either going to happen by diffusion, membrane folding, or transport through transmembrane proteins. When we're talking about diffusion, it's really just that molecules move in a random fashion, but there is a tendency to produce uniform mixtures. It's very simple. Everything wants to move from a high concentration to a low concentration until an equilibrium is reached. So here you can see we're putting in a high concentration of sugar molecules into the water. Here there's still a very high concentration together because diffusion hasn't happened yet. It's just starting. As the molecules diffuse and look for equilibrium, which just means an equal dispersion of molecules, they're going always from a high concentration to a lower concentration. They're trying to spread out and have an even distribution. And this is passive. So no energy is going to be applied to make this happen. It will happen on its own without any supply of energy being provided. That makes it a form of passive transport. No energy needs to take place for diffusion to happen. Okay, when we're discussing selective permeability of a membrane, that just means not everything can come through. There's a list. There's a list of what can come and what can't. Nonpolar molecules can pass through the phospholipid bilayer because they are permeable. They are able to pass through. But that doesn't mean that anything can come through. It's not like we could take any sugar or hydrogen ion or sodium ion and just pass through without any effort. Only certain substances undergo diffusion across the plasma membrane. These molecules are like oxygen, carbon dioxide, and nonpolar lipids. But how are we going to get the other stuff through? It could be with facilitated diffusion. In some instances, molecules that can't diffuse across the membrane can do it with help. And during facilitated diffusion, proteins act as carriers that can bind only to specific molecules and transport them. Transport is going to be limited by the availability of the carriers. And when all the protein carriers are in use, that means that the transport is saturated. No more transport can happen because we have a specific number of carriers and they're all busy. Saturated. That is when transport is saturated. Osmosis is when water moves down its concentration gradient into or out of a cell, and the movement of water is dependent on the concentration of other substances in a solution as well. So the greater the amount of solutes that are dissolved in a solution, the lesser the amount of water molecules that are free to move. You can remember we talked about whenever we had an equal distribution of water molecules and solutes, or just water molecules in a glass, that's an isotonic solution. Isotonic means it is equal, an equal distribution. However, when we have two different sides of a solution and one has more of the substrate, that's going to be the hypertonic part of that solution. The part that has less solute in the solution is going to be hypotonic. I mentioned that you might remember this because when you're hyper, you have a lot of energy, and when you're hypertonic, you have a lot of solute. So the concentration of all molecules or solutes that are dissolved in a solution is called the osmotic concentration. And once again, the isotonic concentrations is if the osmotic concentration of two solutions are equal. If two solutions have unequal osmotic concentration, the solution with the higher solute is said to be hypertonic, and the solution with the lower solute is going to be hypotonic. So we talked about the sodium-potassium pump, and this is such a great example of active transport. These pumps use energy in the form of ATP to pump 3 sodium ions out of the cell and pump 2 potassium ions into the cell. It's also a great example of coupled transport because this is not a process that would be happening if there weren't a coupled reaction that's going on. So if only we had sodium ions that wanted to get out of the cell, the pump is only going to open in one direction and there's nothing to bring it open back in the other direction. So we want sodium out. The sodium attaches with ATP, the pump will open and allow the sodium to exit the cell. Now in that open position, open to the outside of the cell, it's available for potassium to come in and bind. When potassium comes in and binds, the shape of that protein is going to shift once again, and potassium will be brought into the cell. Coupled transport. If we only had potassium working with this pump, it would only be bringing potassium in. We need both. That's coupled transport. And it's active because we're supplying energy to make it happen. Here's our ATP molecule. So, mechanisms for transport across cell membranes. We've got our passive processes like diffusion and protein channels and facilitated diffusion. Even though we've got help happening, there's not energy supplied to make it happen. So facilitated diffusion is still a passive process. Also osmosis. So all of the bringing water into a cell or water going out of a cell is going to be osmosis. And that is also a passive process, meaning no energy has to be supplied. Our active processes. So we've got endocytosis, which has both phagocytosis and pinocytosis as part of it. If you remember, phagocytosis is bringing molecules into the cell. Pinocytosis is bringing liquid into the cell. So it's a very small difference, just two different parts of the endocytosis process. If we're talking about a molecule, it's phagocytosis. If we're talking about a liquid, it's pinocytosis. I mentioned you might remember this by just thinking of Pinot, like Pinot Noir, a kind of wine that some people like. That could be an example that helps you relate to pinocytosis being for the liquid. Also, there are receptor-mediated endocytosis as well. That is an active process that does require energy. And exocytosis. So anytime we're kicking things out of the cell, that is going to be exocytosis. And active transport, just like with our sodium-potassium pump, we've got energy supplied, and that is what is making this protein carrier able to transport things across the membrane. That's our summary of Chapter 4. Now we're on to Chapter 5, where we discussed energy and life. So we discussed there are two types of energy: potential energy, which is energy that is being stored and can be used for motion. And there's kinetic energy, which is the energy of motion. The examples we gave were if you were to have a ball sitting at the top of a hill, it has potential energy. There is stored energy that is not in motion, but it is stored for motion to happen. And then there's kinetic energy, which is the energy of motion. So that is shown during the action of the ball actively rolling down the hill. That is kinetic energy because motion is happening. Also, I mentioned in class, you guys might remember, if I were standing at the start line of a race, poised and ready to go, but not going yet, that would be me with potential energy. Once the sound goes and it's time to race and I'm running, that's me in a state of kinetic energy, the energy of motion. When we're talking about thermodynamics, it's just the study of energy or heat changes. Energy is used and stored by chemical reactions, and chemical reactions are just the making and breaking of chemical bonds. So here's several different vague examples of what chemical reactions could be. It could be combining two things to make a new molecule. It could be breaking a bond to make two new molecules. So we once had this molecule and now we've got this. It could be combining different parts with different byproducts. And these are all examples of chemical reactions. The important part to remember is that a chemical reaction just means you're making bonds or breaking them. When we talked about the laws of thermodynamics, the first law is that the total amount of energy in the universe remains constant. Energy can change from one state to another, but it can never be created nor destroyed. During the energy conversions, some of the energy is lost as heat energy. So the first law of thermodynamics is energy can neither be created nor destroyed. The second law is that the amount of disorder or entropy, those mean the same thing in this case, the amount of disorder or entropy in the universe is increasing. This, we discussed the example of how your bedroom is constantly going to end up in a state of chaos without you intentionally exerting effort to make it a state of chaos, just because you're living, just because the amount of disorder in the universe is always increasing. You're going to have to apply energy to get that bedroom back to a state of being organized and nice and clean. But the disorder is always increasing. When we're talking about chemical reactions, the things that are going into the reaction are the reactants, and the things that are being made are the products. So the easiest example I could find for a visual here is just all of the ingredients that you have to make a cake, and that would be the reactants. And then your product is what you made. So that's what's on the right side of your reaction. That is what has been made by your chemical reaction, your products. And really the same thing's happening here. So I just put these visuals next to each other for that kind of point to be drawn together. So like, wait, reactants and substrates are the same thing? Pretty much. The difference is that we usually use the word substrate when we're talking about reactions that use enzymes, and we usually use the word reactant when we're talking about chemical reactions that don't use enzymes. So when you're talking about what's going into a chemical reaction, if it's about something that doesn't use enzymes, it's usually just going to be referred to as the reactant. When we're talking about things that are using enzymes, we're probably going to use the word substrate. But that's why you see these words kind of interchangeably next to each other, and it's kind of hard to distinguish when to use which one. But that's your clue. If an enzyme is present, you would call it a substrate. If there's no enzyme present in the reaction, you would just call it a reactant. Okay, so on the topic of reactions, we've got endergonic reactions and exergonic reactions. You remember that endergonic reactions have products with more energy than the reactants. These reactions require an input of energy. Exergonic reactions have products with less energy than the reactants. These reactions tend to occur spontaneously. And so really what we're saying is endergonic reactions, you have to put energy in to make them happen. Exergonic reactions, you don't have to supply any energy because they're going to make it. And energy will be absorbed in endergonic reactions. That's why your product has more energy than your reactant, because the energy is being absorbed. And it's the opposite in exergonic reactions because you're releasing energy. So your product will have less energy because it's being released. Also, I mentioned you could remember this by thinking of the exit of energy. Energy is being released. It is exiting in an exergonic reaction. In an endergonic reaction, energy is going into the reaction. This is a great visual too to help remember. Energy goes into the reaction to make something. That's like having to push a boulder up a hill. You have to put energy in to make it happen. An exergonic reaction, energy exits the reaction. So we're not putting the energy in, like we're not putting any energy into making these boulders roll down a hill. It just happens spontaneously in an avalanche. That's a good way to remember the difference here. And here's more of a specific example of what's happening in those situations. So here we've got an endergonic reaction like the breakdown of ATP. So we have energy that is being supplied, and the products are going to have more energy than the reactants. In an exergonic reaction like cellular respiration, energy is produced. So we don't have to put any energy in It's going to be made just by the reaction spontaneously taking place. Here's another great example. So energy goes into the reaction to create products. We've got ATP and a reactant going into the reaction. We are making our product. There's not any energy coming out. It was only put into the reaction. But the product will have more energy than the reactant did because this energy was supplied. And here's our exergonic reaction. We've got energy exiting the reaction. So here comes our reactants together. Energy is produced along with our product. And here you can see them both acting together. So it's not like we ever just have one of these things going on at a time. It's a continuous cycle. Also, we have activation energy to talk about. So all chemical reactions are going to require an initial input of energy, and that is called activation energy. This is the energy that initiates a chemical reaction by destabilizing existing chemical bonds. So it's destabilizing the bonds that you have and making a chemical reaction initiate. Reactions become more likely to happen if their activation energy is lowered. This process is called catalysis. So here's a great little image to kind of bring this point home. We've got an enzyme with an active site ready to bind to substrate. And you can see the enzyme is saying, I'm here to make this reaction faster and easier. It's what they do. The substrate's over here saying like, oh good, I've been looking for my partner for so long. And that's because without the help of an enzyme, the reaction of two substrates that need to come together is going to take way longer to happen. The enzyme's making it faster and easier. Here's our catalysis. We've got our enzyme with our active sites for binding and the substrates that are intending to combine with a chemical reaction. So the enzymes are going to make this happen faster. And that is catalysis, when we catalyze a reaction. When we're talking about biochemical pathways, it's just the concept that the product of one reaction is the substrate for the next reaction until a final product is made. So the biochemical pathway is a substrate is only going to bind to enzyme 1. This specific substrate will only bind to enzyme 1. When it is rearranged or made into a new molecule that is only going to be able to bind to enzyme 2, then a new product comes out that is only able to bind into enzyme 3, and so on until we have our final product. That's a biochemical pathway. Okay, so here's another example of chemical reactions in catalysis where enzymes are lowering the activation energy for a chemical reaction so that it can happen faster. Here you can see an endergonic reaction where energy has to be put in to make a product, and the product will have more energy than the reactant. Here we've got our exergonic reaction where energy is being released along with the product, so the product has lower energy than the reactant. The activation energy is what's making the lowering of the activation energy is what's making this process happen faster and more efficiently. So if we had to reach all the way up to this level of energy to make this reaction possible, to make our product, you can see that that's going to take longer to do than if we are able to lower our activation energy with an enzyme. Also, enzymes need a specific pH. So we can't just have a super acidic pH or have our pH be off and enzymes stay the shape that they are. If you change the pH too much, the shape of your enzyme will change. And if you remember, all of our binding is based on shape. So if you change the shape of your enzyme, now your substrate is not going to bind to it. So here you can see it says, my love, please, it's just a pH change. And there the substrate is saying, no, darling, you've changed. This is a great example because if you change the shape of your enzyme, it makes your substrate unable to bind to it. We also have allosteric enzymes, and they are affected by the binding of signal molecules. So some signals act as repressors. that inhibit or stop the enzyme from functioning when bound. And other signals are going to act as activators that change the shape of the enzyme so that it can bind a substrate. So if we're repressing, that means we're stopping the enzyme from functioning. If we're activating, that means we're changing the shape to make the reaction possible, to make the substrate able to bind to the enzyme. Repressors are shown here, and they disrupt the active site to stop the enzyme function. So you can see a substrate wants to bind to this enzyme and make products. If a repressor comes in, this signal molecule is going to fit just right so that it changes the shape of the enzyme, and now even though there's an opening, this substrate is not able to bind and produce a product. So repressors are, a lot of medications are repressors that will come in and be able to bind and stop whatever reaction would have happened from happening, a repressor. Activators are the opposite. So that means if this enzyme is in its natural shape, a substrate is not able to bind to it. It's like, oh, that's not the right shape. But we bring an activator in as a signal molecule, it binds to the enzyme, and suddenly it reshapes so that now it's the perfect size for the substrate. This is an activator. They're activating enzyme function. Also, we have competitive and non-competitive inhibition that both use repressors. So competitive inhibitors are competing with the substrate for the active site that it wants to land on the enzyme. We've got competition. They're both trying to land. The competitive inhibitor interferes with the active site. You can see the substrate that is intended for the enzyme is like, oh, there's no space for me. It's competitive. They're competing for the same site to land in. Non-competitive inhibitors are also going to stop a function from happening or a reaction from happening, but they're not competing for that one active site. They're stopping the activity without any competing. They have an allosteric site to bind to, and that is where the enzyme is going to change shape so much that the substrate will no longer be able to bind. So even though they're not heading for the same destination, because this non-competitive inhibitor that's a repressor bound to the allosteric site, that changed the shape of the enzyme enough that now this doesn't fit like a puzzle piece the way it should. So this is another, I use this same image twice because both of the example of a pH change and an allosteric binding site being bound to are going to change your enzyme shape. So in this situation, we're made for each other, please don't go, and the substrate says, no, darling, you've changed. That's because we've got something bound to the allosteric binding site that will change the enzyme shape. Now we need to talk about ATP again. So as you remember, ATP stands for adenosine triphosphate. Adenosine is just meaning adenine, our nitrogenous base, and a sugar, which is ribose. So that's our adenosine of the name. And our triphosphate is for 1, 2, 3 phosphates that are on this molecule. You can see this note here. It says this phosphate is ready to go. It's like, just say the word and I will break this bond. This phosphate is poised to break off, and it's because it's a reaction that's happening constantly. We're constantly breaking off a phosphate of ATP molecules, and then in the next process we'll be adding that back on. So we go from ATP to breaking off a phosphate so that we have ADP, adenosine diphosphate, di is in 2, and then that process is going to constantly go back and forth. The important part to remember is we've got 3 phosphates. One is poised to break off at any moment. We've got our nucleic, or our nitrogenous base and our sugar. 3 phosphates. When the endmost phosphate group is broken off, an ATP molecule energy is released. So you can see we've got ATP, we've broken a phosphate bond, so now we have ADP with 2 phosphates, and we've got an inorganic phosphate, a phosphate ion, and energy has been created. Well, energy has been supplied. The phosphate bond breaking frees up a phosphate ion and provides energy as well as the newly formed ADP molecule. So you can see we've got ATP. We break that phosphate off. Here's the phosphate that is no longer part of it. Here's the 2-phosphate molecule. Once we've got ATP breaking that phosphate off, energy release happens. That's how energy is supplied. Now we have ADP molecules. There's free-floating energy that will add to those ADP molecules and phosphate ions to reattach a phosphate to ADP, and now we have ATP. This is a continual cycle. So this is going to happen back and forth over and over, and it's going to power all cellular function. Cells use 2 processes to convert energy from the sun and from food molecules into ATP: photosynthesis and cellular respiration. In photosynthesis, cells convert energy from the sun into ATP and then use it to make sugars that store potential energy. In cellular respiration, cells will break down the potential energy in sugars and convert it to ATP. So photosynthesis, we are converting energy to store it as sugars. And in cellular respiration, we are breaking down the potential energy in sugars and converting it to ATP. In chapter 6, we learned about photosynthesis. In photosynthesis, all of the energy is used by almost all living cells will ultimately come from the sun. Photosynthesis is the process of harvesting that energy and making it usable. So plants, algae, and some bacteria are able to capture the sunlight energy by a process called photosynthesis. And only about 1% of the available energy in sunlight is actually incorporated into organic compounds during photosynthesis. But it still makes everything possible. So how do leaves take in carbon dioxide and release oxygen and water vapor? Leaves are able to do this by having openings that are on the surface of the leaves, on the underside of the leaves. They're called stomata. So when the stomata are open, that allows exchange. And whenever they're closed, we're allowing the release of oxygen and water vapor. So it's just to take things in and out. Plants use these openings for gas exchange. Stomata. Here we have a photosynthesis example as a reaction. So we have our reactants that are going in, which will be carbon dioxide and water. We know that plants take in carbon dioxide. We know that they take in water. We know that they take in the energy from the sun. And with those things, through photosynthesis, they're able to create sugars and a byproduct of oxygen. So their goal is sugars. That's what they're storing for food. That's the goal of their process. The byproduct that we benefit hugely from is the fact that they make oxygen as well during this process. And these are our products to the reaction, what is made, like the cake. The pieces that go in and the pieces that come out. So photosynthesis takes place in 3 stages inside the chloroplasts of plant, algae, and some bacterial cells. The 3 steps to photosynthesis are the capturing energy from sunlight, and then second, we have to use that captured energy to produce ATP and NADPH. You don't need to know that it stands for nicotinamide adenine dinucleotide phosphate hydrogen. That's irrelevant. I included that in case anybody was like, wait, what does NADPH stand for? Well, that's what it stands for. We're always going to refer to it in this condensed way, NADPH. And it's just a useful molecule that's going to then be used in the next part of photosynthesis. Step number 3, where we're using the ATP and NADPH to make the carbohydrates from CO2 in the atmosphere. So those are our 3 basic steps to photosynthesis, and it's all happening inside the chloroplasts. So we've got our leaf cells. Here's a close-up of our leaf cell with our nucleus, our central vacuole, the chloroplasts scattered throughout, You can see the cell wall. When we zoom into these chloroplasts, that's where all the activity is happening. And you can see we have these structures called thylakoids. When you stack them together and have one stack of them, that's going to be referred to as a granum. And then, much like cytoplasm, there's this fluid-like aqueous liquid inside the chloroplast, and that's called the stroma. So all of our photosynthesis processes are either going to be happening in the thylakoids or in the stroma. Thylakoids is where our light-dependent reactions are taking place. So if the reaction requires light, it's happening in the thylakoids. If it is going to happen, if the reaction is going to happen regardless of whether or not sunlight is there, then that's a light-independent reaction, and it's happening in the liquid called the stroma. So when we're talking about that process where we have light-independent reactions happening in the stroma, that's going to be the Calvin cycle. The Calvin cycle is what we're talking about for the part of the process that's taking CO2 along with the ATP and NADPH that has been made from our light reactions. So CO2 is going into this ATP and NADPH. Our products are NAD+, a phosphate, an ADP, and sugar. Sugar is the main goal. So the Calvin cycle is what's happening inside the chloroplast to produce sugar. Again, we have byproducts. So here are byproducts of that process. They are going back into the thylakoid to be used for the other part of the photosynthesis, the light-dependent reactions. You see the light here. The thylakoid depends on the light for this reaction to be possible. It also needs water. So we need light and we need water. We need the byproducts that are happening from the simultaneously occurring Calvin cycle that's ongoing. And that is going to make it possible to produce ATP and NADPH, which is needed to make the sugar. So these are two parts of the process that really feed into each other constantly. A byproduct of our light-dependent reactions is oxygen. So like I said, we're not trying, the main goal of the plant cell is likely just sugar to have food. But as a result, it's going to be expressing oxygen because this H2O has been broken down. The energy to power the Calvin cycle's production of sugar comes from the light-dependent reactions in the thylakoid. So I just circled this kind of roughly just to help you focus in on that point. Energy that is needed for the Calvin cycle to happen is being made by the reactions happening in the thylakoids. And that is also a result of chemiosmosis. So chemiosmosis is the process used to move protons up a concentration gradient for production of ATP. Chemiosmosis produces ATP. And that is happening in the thylakoid membrane, so the membrane that surrounds the thylakoids that are stacked on top of each other. That is all. That is a huge, fast summary of some pretty intense chapters. But if you study this material, you can do very well on this test. You can do this. Thanks, guys.